Well, hello, everybody. My name is Emilio Bruna, and I am at the University of Florida and also president of the ATBC. And I am thrilled to be here with Javi Rudolph, who's our tech person running the Zoom here. And the two of us are helping to present to you here this session on lightning talks and biotic interactions in the face of global change. Thank you so much for joining us. We're really excited about this. It's been a really wonderful couple of days. I hope you're energized and excited by what you've seen so far. We're going to have a great session here. I'm going to quickly introduce our speakers, and then I'd like to go through a quick bit of logistics about the best way to ask questions. You've probably already heard this in other sessions, but it never hurts to repeat it. And then we'll head into the presentations. Does that sound okay? Okay, so our first, our order of speakers today, our presentations, as I have them so far, are going to be Flavia Nogueira de Sá from the University of Brasilia in Brazil, Antonio Crespo from the Universidad de La Salle in Ecuador, Sarina Thiel from Philipps University Marburg in Germany, Danielka Tilakarathni from the Department of Zoology at, University of Peradeniya in Sri Lanka. Anoma Pereira, also from University of Peradeniya in Sri Lanka. Biswan Mahapatra from the Ashoka Trust, A-Tree in India. Anne-Marie Gal from Iowa State University in the United States. Aimer Vasquez from Universidad del Valle in Cali, Colombia. Biruk Ayaleo from Stockholm University in Sweden. And Valerie Millisey from the University of Connecticut in lovely Storrs, Connecticut. Thanks to you all. Really excited about this. Stick around. We're looking forward to your presentations. If you have any questions, please be sure to submit them via the Q&A function on Zoom. If those questions come up during the session, if you have questions afterwards that come to mind that you'd like to ask, please do post them. Post them on the Whova page for this session, and that way speakers will be able to return to them, answer them after the session is no longer live, and other people will be able to see them as well. So anybody else who comes back to see the talks will be able to check them out. And so with that, I'm going to mute my microphone, turn off my camera, and let's see some science.
Hi, my name is Flavia. Today I'm going to be talking about my work on the influence of phytochemistry on herbivores. We know that host plants presents a high diversity of secondary metabolites, and they are influenced by ecological interaction that drive this complex mixture of substances. And we also know that the production of these metabolites is highly seasonal, due mostly to environmental factors. We conducted our study in this very seasonal environment, the Cerrado, the Brazilian savanna, well known by the regions of species and very important by the high degree of threatening. The seasonality in Cerrado is represented by the dry months of the fall and winter months of the south, and by rainy or wet season in the spring and summer months. We studied caterpillars which are known in the area by their seasonality and also by the turnover of species across the different climatic seasons. So to verify the seasonal plasticity of the plant metabolism at its specific level and to determine if these patterns are consistently expressed in different species, we chose 12 host species, which are quite abundant in the area. We collected caterpillars from 2011 to 2015 and organized them in 4 communities, those collected in the months of the rainy season, those collected in the months of the transition between rainy and dry season, those collected in dry season, and those collected in the transition between dry and rainy season. For the chemical defense investigation, we collected leaves in 2017. These leaves were collected in the same area where the caterpillars were previously collected. We collect them 4 times in the year, representing the 4 climatic seasons. The methanol extracts were submitted to hydrogen nuclear magnetic resonance. So we took this chemical structural complexity approach and not focusing on any substances as most of the works do. We collected 224 species of caterpillars. About 50% of them were singletons. So we decided to study the most abundant caterpillars, and we found that most of the species was very specialized in the diets, and none of them was found in a single season. We found this high turnover of species in the different communities, represented by the low similarity, 50% or less in most of the species. And we found that the communities were mostly organized by their host plant species and not by the seasons in which they were found, with a few exceptions, but always with very low similarity. We found a similar pattern for the phytochemistry, finding that the similarity was higher between samples of the same species and not in the same seasons. This difference in similarity was basically explained by the chemical shifts that they vary across different seasons. And you can see here, some chemical shifts are more abundant in some seasons, not the other. Sometimes it disappear. And this is reflected by the difference in the samples across different seasons, especially in the dry and the wet season samples. This is reflected in consistent reflected in most of the species that we found, and we can see here the most important difference between the samples of the wet and of the dry season. We compared the similarity matrix of caterpillars and phytochemistry, and we found a quite high relationship. In fact, when we looked for those most abundant caterpillars, we found that although any of them was specific to season, most of them were found more frequently in some seasons. So we can suggest that these caterpillars were looking at some specific chemical functions that are found in their host plants. So for the next steps, we'd like to correlate these caterpillars to chemical functions. And also you want to identify these chemical functions and to check how they vary across different seasons. I would like to thank students, colleagues, financial agencies, and you for your attention.
Hello everyone, my name is Antonio Crespo. I am an associate professor at Universidad de la SOAY in Cuenca, Ecuador. This will be a summary of a manuscript that was accepted for publication in the Journal Restoration Ecology, entitled Key Plant Species to Restore Plant Hummingbird Pollinator Communities in the Southern Andes of Ecuador. The tropical Andes are a biodiversity hotspot, but at the same time one of the regions most threatened by deforestation and degradation. So restoration is urgently needed in the region, but most projects focus only on structural elements of ecosystems and disregard key ecological processes such as pollination. So this process is paramount considering that at least 87% of all flowering plants are pollinated by animals. So a better understanding of plant-pollinator interactions may be critical for restoration. Network ecology can be especially useful in this context. For example, interactions in pollinator networks tend to be concentrated in a few plant species that receive most pollinator visits. These plants increase functional redundancy, making the network more resilient to change. Thus, these plant species can be reintroduced in a degraded landscape to attract and sustain pollinators. Here we focus on the community of hummingbirds and the pollinated plants. But why hummingbirds? Well, more than half of all angiosperm families use hummingbirds as pollinators. Hummingbirds also carry pollen loads over long distances and can be active in harsh environmental conditions, thus making them perfect candidates to enhance gene flow at a landscape scale. The objectives of our study included the following: which plant species are key for hummingbird pollinators in different vegetation types, and how are functional traits of these plant species associated to centrality values. We'll talk about centrality values a little bit later in the talk. Our work was conducted in the Southern Andes of Ecuador in 4 independent reference sites, each with a distinct vegetation type. Our study sites were either surrounded by or within an agricultural landscape matrix. Now, vegetation types in our study sites were old-growth forest, secondary forest, hedgerows within pastures, and mountain shrub. For our flower hummingbird surveys, we conducted 16 observation periods between February 2012 and October 2015. We recorded the frequency of legitimate hummingbird visits to plants by either direct observations or using a video camera. In each vegetation type, we placed 3 plots of 200 by 5 meters. Now, for our functional traits, we chose a series of traits that are known to significantly influence pollination interactions. You can see them here on the screen. Now, for each site, in each survey period, in each species, we calculated 3 commonly used centrality indices that you see here on screen. Then we used a principal component analysis to obtain a global measure of centrality that combines the information of the indices that you see on screen. So we call this a centrality index. So plant species that have positive values of the centrality index were considered key species, and those with negative values were considered peripheral species. To analyze how functional traits were associated with the values of the centrality index, we used multivariate linear mixed models, as you see on screen. For our results, we found a total of 123 unique pairwise interactions between hummingbird species and plant species, which included 44 plant and 15 hummingbird species. For each vegetation type in each observation period, we identified key plant species with positive values in at least one observation period. Those are the blue dots. In all forests, we found 11 key plant species, 7 in secondary forest, 11 in hedgerows, and 4 in mountain shrub. Now, the interesting thing here is that species roles are dynamic across time. So the same species may be peripheral in one observation period and key in another. We found that flower abundance and life form were the traits with the most influence on the centrality value. These findings suggest that hummingbirds can be highly opportunistic using any shrub species with abundant flowers that are available in the environment. Now, the centrality metrics used in this study are a cost-effective and ecologically robust method to select plant species for restoration, as opposed to relying on local expert knowledge on very costly and time-consuming trial and error experiments with dozens of species. Our method, on the other hand, resulted in a small set of key plant species that could be used for restoration plantings. However, in most cases, key plant species are not available in local nurseries, so this highlights the need to develop propagation protocols and to study responses to different field conditions urgently. And with this being said, thank you very much for your attention. I hope to see you all very soon.
Hello, my name is Sarina Thiel and I'm currently a PhD student at the University of Marburg. And now I'm going to talk about vertically stratified frugivore community composition and interaction frequency in a liana species fruiting across forest strata. In forest ecology, the vertical organization of structures, microclimate and biota has been a recurring research topic since the 1950s. Especially in tropical forests, vertical stratification is a widespread phenomenon for plant and animal communities and a key factor for structuring biodiversity. The observed vertical stratification of many vertebrate frugivores in terms of abundance, richness, and community composition also affects associated ecological processes, such as frugivory and seed dispersal. For instance, Schleuning et al. Reported that the degree of specialization in a plant frugivore network in a Kenyan rainforest is vertically stratified, probably due to differences in foraging behavior among animal species that differ in their dependence on fruit in their diet. Further, vertical stratification was shown to play an important role in the diversification of plant and animal traits. However, it is still unclear whether this vertical structure is driven by differences in plant community composition among strata, or rather by inherent preferences of frugivores for specific strata. To date, all studies investigating vertical stratification of plant-animal interactions focused on plant communities, where each plant species presented fruits within a single stratum. This implies that species in stratum are confounding variables. The neotropical liana, Macravia longifolia, is an ideal study organism to investigate the vertical stratification in plant-frugivore interactions, as it provides fruits from the understory to the canopy. This enables us to investigate changes in frugivore community composition and interaction frequencies across forest strata for one particular fruit resource, independent of changes in fruit resources of this specific species. We hypothesize that frugivores preferentially forage in a distinct vertical niche, even if the same resources are available along the entire vertical gradient. Obligate, opportunistic, and partial frugivores are hypothesized to differ in their relative importance for a particular stratum. And lastly, we hypothesize that functional traits of birds play an important role in determining their vertical foraging niche. During 418 observation hours, we recorded fruit removal from 24 Magravia longifolia individuals across forest strata. We recorded 862 interactions with 43 frugivore species. Those were assigned into guilds according to dietary specialization. And for birds, we further determined 4 morphological traits. We found that frugivore visitation, diversity, and community composition significantly differed among strata, with a high interaction frequency of a few partial frugivore species in the understory and a low interaction frequency of many different obligate frugivores in the canopy and midstory. Partial frugivores were the most abundant species in our community. Opportunistic and obligate frugivores were less frequent visitors. Whereas partial and opportunistic frugivores foraged across strata, obligate frugivores were predominantly found in the canopy and midstory. Further, avian traits differed among strata. Avian frugivores foraging in the canopy were mainly large-bodied species with pointed wings, whereas under and mid-story avian foragers were smaller with more rounded wings. So, avian frugivores remained within their preferred vertical niche, even though an attractive food resource was available across strata. Vertical stratification appears to be an important driver of a species interaction niche and is associated with its morphology. Hence, the niche differentiation of species along the vertical forest gradient may be a key factor promoting tropical forest diversity. Losing this vertical structure due to anthropogenic disturbances such as selective logging of canopy trees or the simplification of the vertical structure in agroforestry systems and forest regeneration areas would imply a loss of tropical diversity and thus needs to be carefully considered in tropical forest conservation. With this, I want to thank you for your attention, the DFG for funding this project, and all people who contributed. Thank you.
Hello everyone, today I am going to share quite interesting facts regarding invasive species. Do you think that exotic gastropods can invade natural forest system? So this presentation will answer that question. Sri Lanka is home to 18 species of exotic gastropods. Most of them are agricultural based. Not only that, they will establish well in upcountry agricultural lands, where most of the lands are closely located to the natural forest. This is where this problem arises. These exotic can easily encroach this natural forest. If they invade this forest, it is a huge threat to the native species. Therefore, I want to evaluate the forest invasion of the exotic gastropods in the Nuwara district, Sri Lanka. After preliminary survey, we selected 60 random forests proximately to the agricultural lands. This survey was conducted from 2018 to 2019. Each site was established 10 to 51 square meter plot for surveying the gastropods. And this study was conducted during the daytime. The density and the environmental correlations was analyzed using this data, as well as we have prepared species distribution maps using ArcGIS. So during this 1 year period, we were able to encounter 410 individuals belonging to 8 species, including 1 exotic slug species, that is Deroceras leve, and 7 exotic land snail species. So most abundant species during this survey was Bradybina similaris, Lissacatina, and Alopius. The most interesting fact in here is most of these exotic gastropods were highly abundant in forest buffers as well as the marginal vegetations between agricultural lands and the natural forest. So again, these exotic species show a wider range of tolerance for most of the environmental factors we measure, such as temperature, elevation, relative humidity, rainfall, as well as the soil pH. So because of this wide range of tolerance, these species can successfully invade the natural forest system, and they can easily compete with the native species for food, shelter, and the niche. We were able to find that most of the exotic snails are found in mid elevations, where the moderate temperature and the slightly acidic soils are present. And on the other hand, the exotic slugs were found at higher elevations and low temperature and acidic soils. So these facts are very important when it comes to the controlling strategies. So this diagram will show how these exotic species arrange according to the measured environmental variables. So in here you can see two main clusters of the exotic gastropods. So this first cluster will majorly governed by the rainfall, and the second cluster including Bradybina, Agellustra species, they were mainly governed by the elevation fact. I hope you will get an answer for my question at the very beginning. Yes, exotic pest gastropods can invade the tropical montane rainforest. We found that globally invasive species such as giant African snail, Bradybina similaris, and Deroceras species were successfully invade our natural forest due to the climatic changes and the anthropogenic activities. They are a huge threat to the native species in this natural forest. Therefore, this data, the population data, environmental data, and the distribution data can be used to detect early measures as well as to establish well-controlling methods of these exotic gastropods being stopped becoming invasive species. This study is not only important for the Sri Lanka, but also it is really important for the other tropical rainforest in the world. So we can use this data to formulate a proper management action to controlling these pests and conserve our native species. Thank you.
"47 alien exotic plant species that grow in various ecosystems of Sri Lanka have been identified as serious invaders. We need more information pertinent to the actual risk or the potential threats imposed by these species when taking decisions to control them. Therefore, we conducted a comparative study to investigate the allopathic effects of common invasive plants of Sri Lanka using a seed germination bioassay. I am Anoma Pereira. The species selected for this study include Alstonia macrophylla, Panicum maximum, Clusia rosea, Ostripaturum minulifolium, Lantana camara, Clydemia hirta, Calliandra calothesis, Tithonia diversifolia, Prosophis duliplora, Clerodendron cordilocolari, and Mimosa pigra. We investigated whether the leaf and root extracts of these species would inhibit the germination of mustard seeds or delay their germination under laboratory conditions. We prepared 1, 5, and 10% dilutions of fruits and leaf extracts of these plants. Distilled water was used in the control experiment. We soaked 100 mustard seeds in each prepared solution for 24 hours, kept on tissue layers moistened with respective extract in 5 replicates, and observed these every day for 5 days. We recorded the cumulative number of germinated seeds, and at the end of the 5th day, we measured the length of the hypocotyl and the radicle of germinated seeds. Our results showed a significant difference in percentage seed germination in extracts of different invasive plant species. This slide compares the suppression of seed germination in 1% root extracts with that of the control experiment. Here the y-axis is percentage ungerminated seeds, and this is the control. Several species inhibited seed germination up to about 55 to 65%, but some other species appears to be exerting a lower suppression. Clydemia hirta and Clusia rosea exerted a very mild inhibition. All the species except Lantana camara, Clydemia hirta and Clusia rosea delayed the time taken for germination. Germination of mustard seeds in different leaf extracts is also more or less the same as the patterns that we observed in root extracts, but leaf extracts always exerted a milder impact compared to respective root extract. However, Clydemia hirta and Clusia rosea leaf extracts did not inhibit germination of mustard seeds. Instead, this dramatically enhanced the initial seed germination. Leaf extracts of the rest of the species inhibited seed germination, and all these except Ostriopetorium inulifolium delayed the time taken for germination. We observed species-specific responses, especially when increased the concentration of leaf extracts from 1% to 10%, but in Prosopis duliflora, increased concentrations of both leaf and root extracts dramatically increased the inhibition of seed germination. We observed higher values for average hypocotyl and radical lengths in all experimental treatments compared to the control, indicating no or minimal adverse impact caused by these extracts on the initial growth of germinated seeds over 5 days. Therefore, we conclude that all the examined invasive species may possess allochemicals which increase their invasive potential by suppressing seed germination. The degree of seed germination inhibition may vary with the invasive species, the plant part, and the concentration of allochemicals in the surrounding. Leaf extracts of some invasive species that listed as risky invaders in Sri Lanka do not necessarily suppress seed germination. Instead, these may enhance the seed germination. However, further investigations are required to explain potential threats exerted on native species by these plants. Thank you.
Hello, my topic is distribution and determinants of animal invasive species. Look at the fish, isn't it beautiful? This is lionfish, which has been transported for aquarium across the world. This is native to Indian Ocean, but found in West Atlantic too. So what's the big deal? Its population grows rapidly. Females produce up to 30,000 eggs a month, each of which will hatch in 4 days. It feeds on native herbivores, which causes unregulated algal bloom. Therefore, it imbalances the coral reef system. Lionfish are venomous with these spines containing apocrine-type venom glands, which can cause mild fever, nausea, even to death. Lionfish are concerned to coastal managers due to their potential threat to fisheries resources, native fish communities, and human health. So, this is an invasive species. Similarly, there are lots of invasive species. Here we can see how invasive species have adverse impact on biodiversity, ecosystem services, economy, and human health. For better management, we need to know their distribution and how the factors which are responsible for their introduction. Like we saw in case of lionfish, in which alien species are defined as species introduced by anthropogenic factors outside of their natural geographic range and which are capable of sustaining a self-replacing population. They are one of the major threats to biodiversity. In 1958, Charles Elton had published the first systematic study on biological invasion, that is, the ecology of invasion by animals and plants. In this study, I have focused on animal invasion across the globe as they are less studied compared to plants. So, I'm looking into understand the patterns and distribution of animal invasive species at global scale. I'm using mainly three types of data related to species, environment, and socioeconomic factors. So, species data mainly consist of species list and occurrence data which have been extracted from open sources like GISD, GBIF, and CABI. Environmental data and socioeconomic data have been collected from WWF, GIS, CDAC, World Bank data, FAO, etc. Here, the thicker line represents most number of invasive species introductions, and that is the case is in between North America and Asia. The skewed maps shows number of invasive species per unit area for each country. Most of the European countries and islands have higher number of invasive species than the other parts of the world. However, US has the highest number of invasive species. In case of biomes, temperate broadleaf and mixed forest is most vulnerable biome to invasive species. However, for vertebrate invasive species, it is tropical and subtropical moist broadleaf forest. Similarly, Palearctic being the most vulnerable realms to invasive animals. Invasive species causes homogenization. Here, clumps shown in different colors contains different invasive species from different realms all together. All realms like Nearctic, Neotropic, Palearctic, Australasia, Afrotropic, Indomalayan. So clumps are countries with similar geographic regions. Structural equation modeling shows how invasive species are related to various socioeconomic factors. For example, human capital index, trade, export, import, agricultural land are related to invasive species. GLM analysis shows the major drivers of invasions are being railroad, container ship, trade, and human capital index. Number of invasive species and number of threatened species for each country are correlated. It means a country with higher invasive species will likely to have higher number of threatened species. So, developed countries are having more invasive species than developing countries. We need a better documentation of invasive species data for developing country. Temperate regions are more prone to invasive species than tropical regions, which can be an indicator of climate change. Invasive species put pressure on threatened species and human activity facilitates invasive species introductions. I would like to thank Dr. Arvind, Dr. Ravikant, Atri, Mahai and their support, and I would like to acknowledge DST SDSN for funding. Thank you.
Thank you for coming to my virtual talk. My name is Anne-Marie Gowell, and I'll be talking about perceptions of invasive species and control efforts in a novel island ecosystem. Why should we be concerned about how people perceive invasive species? Well, here are a few examples of conservation efforts that have been thwarted because of community concerns. Control of feral and outdoor cats usually arises concerns amongst people who care for these charismatic creatures. Control of aquatic weeds using herbicides, for example, in waterways may raise public health concerns. And cutting down aesthetically pleasing trees to prevent invasive bark beetles from spreading could be a concern amongst residents who appreciate these trees in their neighborhoods. In this talk, we'll look at the perceptions of invasive species in Guam, a small island in the Western Pacific. Our most infamous invasive species is the brown tree snake, which is responsible for the extinction or extirpation of all of our native forest birds. To study Guam residents and their perceptions of invasive species and their environmental threats, we recruited small discussion groups. When recruited, we asked them about local environmental threats, including invasive species and how they were managed. These results, mostly encapsulated in direct quotes from the transcripts, were determined using grounded theory to extract major consistent themes amongst the groups. Our first major takeaway was that people seem to be more concerned with coconut rhinoceros beetle, a relatively new invasive species, compared to brown tree snakes. It was mentioned in more groups and was ranked more highly when people were asked to rank their species of most concern. My house, all the coconut trees are destroyed, but of course we grew up with the brown tree snake, so it's not so much an invasive species for us. Second major takeaway is that there were marked differences between the way older participants and younger participants discussed these issues. Older participants had more general knowledge about environmental threats and the environment, but they also had less trust of environmental agencies and expressed more concern about certain types of treatments, especially those involving toxicants or chemicals. Another interesting finding was that some of the older residents doubted the snake's involvement in the bird disappearance, and in fact attributed it to pesticide spraying that occurred when thousands of Vietnamese refugees were housed in Guam temporarily at the end of the Vietnam War. Finally, questions arose among older residents about costs and effectiveness of current efforts, such as the current effort called the aerial toxicant drop, which involves lacing dead neonatal mice with acetaminophen, which is 100% toxic to snakes, attaching these mice to streamers so they will stream up in the trees where the snakes feed, and then shooting them out of the back of helicopters. It just seemed like so much money went into each one of these drops. And some people doubted their effectiveness. I don't think it would work at all. The problem is that they probably dropped a few million rats, and it wouldn't even put a dent, probably, in the snake population. So what can we come away with from these discussions? First, it seems like there's a lot of work to be done to regain trust, especially among older residents. Younger residents seem to have less knowledge about all of these issues, including naming invasive species and which ones they are most concerned about, as well as knowing anything about the control methods that were currently being employed or planned. And so these knowledge gaps need to be addressed, but we also need to foster trust with the younger residents who seem to have more faith in our local environmental agencies. And finally, we should treat communities as partners and trusted sources, recognize their lived history. There were toxicants that were sprayed in Guam, and these environmental concerns should absolutely be addressed in addition to any other ecological problems that we're trying to address today. Thank you very much.
Hello, I am Aimer Vázquez from Universidad del Valle. This presentation is part of my PhD thesis carried out at the Universidad del Valle in Cali, Colombia. The beetle represents the 18% of the diversity of Eukaryota, with the weevil as the most diversity family. This specific reason has been associated to the diversification of the angiosperm plants. An important step to generate this diversification is the insect colonization, as it produces new interactions. The persistence of the interaction requires a relative stability of both parties in a geographical context. The mass death of peach-palm crops in the last decades, caused by the Dynamys boracia and Rhynchophorus palmarum in Colombia, is an excellent opportunity to document which factors favorite a new insect-plant interactions. because this crop is present in the variable's environmental condition, making it easier to evaluate in the field how this condition favor the interactions. So we explore the following three questions. How is the new interaction occurring? What environmental factors affect this interaction? What characteristics of the weevils are favoring the permanence of the interactions? We visit 38 fish pulp production sites in Colombia. In 32 of these, a variable area transit was implemented to document the infestation levels. All the crops presented damage, with the exception of some localities in the southwest of the Amazon. In average, the Pacific had the highest infestation. 4 stages of damage were detected, starting with the attack of the voraciety of the unopened inflorescence, followed by the perforation of the stem by both species until dropping the crown. Insect identification was based on the morphology and sequences of koi genes. In green and bluish green underlining, you can see the sequences for weevil of the fish pulp. For the second question, we associated the infestation level of the 32 fish pulp crops with 109 variables of the local management, soils, landscape, and climate. The landscape variables were the most important according to the separate PLS GLM models. The fractal dimension index and number of the patches of the non-forest cover were more associated with the level of the infestation when all the significant variables were analyzed together. These variables are associated with the forest fragmentations. This pattern could be explained by the fragmentation effect in the density of the natural host palm, Anocarpus batawa of Divoracea, in forests close to the fish pulp crops. Finally, the adult weevils from this study and from the entomological collection were studied with a linear morphometric analysis according to host plant, measuring 17 body parts of the 22 adults of Divoracea and 108 of R. palmarum. In Divoracea, one individual from a natural host was different from those of the fish pulp, regardless of the geographical regions. The addition of the other specimens with unknown hosts demonstrate that the only 2 weevils collected from the Pacific region before the massive death of fish pulp were clustered with the adults of the natural host. Body size was the most important relevant variable in the PCA analysis. Likewise, R. palmarum, the individuals in sugarcane sample were different from those in 2 species of pulp trees, even within the same region. Body size was the most relevant variable in PCA analysis, which provides significant differences. These weevils are very likely to modify the length of their life cycle, as suggested for R. ferruginous in date pulp varieties. The development of koi genes phylogeny confirmed that 8 R. palmarum samples for sugarcane were different from fish pulp samples in the Pacific. In conclusion, this new interaction may be mediated by 3 factors: landscape, alternative natural hosts, and life cycle duration. We thank the Minister of the Science of Colombia for the funding, as well as the other institutions and researchers for their collaboration and support. Thank you very much.
Hi everyone, my name is Broka Yaliu. I'm a PhD student at Stockholm University. Today I will present you one of my projects, which is entitled as the impact of shade tree identity and the canopy cover on coffee pest and the diseases. Okay, as introduction, coffee naturally grown under the canopy of different shade tree species. As you can see here, these are some of the shade tree species where coffee are grown under their canopy. These shade tree species may also differ in terms of crown architecture, and this difference in crown architecture may affect the quantity and the quality of light that penetrate their canopy, and this difference may affect the microclimate, especially the temperature or relative humidity under the canopy. Trees also, they are different in terms of nitrogen fixing ability. Some of the shade tree species have the capacity to fix nitrogen because of the association with nitrogen fixing bacteria, and some of are not. Overall, these things may shape the diversity and the composition of pests and the diseases that live under the canopy. So my first question is, do levels of insect pests and diseases vary in relation to shade tree species identity and the canopy cover? And then my second question is nitrogen fixation drives the effect of shade tree species identity and the canopy cover on coffee pest and the diseases? Okay, to answer these questions, we conducted two complementary survey in commercial coffee plantation and the landscape survey in southwestern Ethiopia. In commercial coffee plantation, we assume that the environment is more or less homogeneous, so it is easier to identify the effect of shade tree species. But patterns which is uncovered in the more homogeneous environment may not be reflected in heterogeneous environment. Therefore, for this, we conducted a large-scale survey in 58 coffee growing sites along the management and elevation. Therefore, in addition to that, we also collected that different kinds of pest and disease data. When I come to the result part, this is the impact of shade tree species on coffee Plush minor, and this cross box plot showed that the tree species which fix nitrogen, and the shade tree species have no effect on coffee Plush minor at commercial plantation study. But in the large-scale survey, it showed that coffee Plush minor had, or shade tree had, an impact on coffee Plush minor, specifically on a coffee tree which is grown under Acacia abyssinica, and Ficus vasta had highest incidence of coffee Plush minor. This is the impact of shade tree species on Antestia bug, and the Antestia bug is not affected by shade tree species on both studies. This is the impact of shade tree species on coffee leaf rust, and on both studies, shade tree species had strong impact on the incidence of coffee leaf rust, and the highest coffee leaf rust infection was observed on a coffee which is grown under Acacia abyssinica and Croton mastitis. This is also the impact of shade tree species on coffee berry diseases. Similarly, here also shade tree had a strong and a variable effect on coffee berry diseases. And at the high level of coffee berry disease was observed on a coffee shrub grown under Acacia abyssinica and Polances fulva. Okay, when I come to the impact of canopy cover, this is the impact of canopy cover on coffee Plush minor. As you see here, canopy cover have no effect on Plush minors at plantation study, but coffee Plush minor negatively affected by canopy cover in the landscape survey. This is the impact of canopy cover on Antestia bug, and as you see here, the presence of Antestia bug negatively affected when the canopy cover increasing at both studies. This is also the impact of canopy cover on coffee leaf rust study. As you see here, we found impact relationship between the coffee leaf rust and the canopy cover at commercial study, but we didn't find any relationship between the coffee leaf rust and the canopy cover in the landscape survey. We also found no relationship between coffee berry disease incidence and the canopy cover at landscape survey. Okay, as a summary, in general, shade tree species left weak effect on insect pests, whereas the canopy cover strongly affected the insect pests. In contrast to insect pests, the coffee leaf rust and the coffee berry disease differed among the shade tree species, but this difference could not be explained by nitrogen fixing ability of the trees. In general, our finding helps to understand the variation in pest and the disease dynamics in natural forest and agroforests. And it also may inform the selection of shade tree species by coffee producer. Thank you for listening.
My name is Valerie Malisi, and I will be talking to you about patterns of pathogen attack on seedling communities across a neotropical precipitation gradient. Many studies have found interactions with fungal and oomycete pathogens alter seedling outcomes. However, these studies usually look at a subset of the seedling community, or they remove pathogens via the application of fungicide or soil sterilization treatments. I was interested in going into the forest to observe naturally occurring seedling communities to look for some potentially important patterns in plant pathogen interactions. First, I was interested in if there was a positive relationship between mean annual precipitation and pathogen risk as predicted by Gillett and Gibnish. Additionally, I was looking for evidence of negative conspecific density and distance dependence consistent with predictions of the Jansen-Connell hypothesis. And next, I was looking for any potential traits that could explain these relationships, such as seed dispersal mechanism. To do this, I surveyed 5 different forest sites located across a precipitation diversity gradient in Panama. I surveyed each site 5 different times, and during each visit, I would perform 10 unique transects. These transects were 10 meters by 1 meter, and along each transect, I would identify seedlings to species and then classify them as either symptomatic or asymptomatic of pathogen infection. I observed in total 71 different species, and I found that disease was actually quite rare, or observable disease was rare among seedlings. Only 8% of my seedlings were symptomatic of disease. Additionally, most of my symptoms were caused by foliar pathogens, although I was looking for both damping off and foliar disease. So all of my results will be in the context of foliar pathogen damage. I did find that there was a relationship between mean annual precipitation and pathogen risk. However, it was opposite to my hypothesis that it would be a positive relationship. In fact, seedlings in the wettest sites were less likely to be diseased than seedlings in the driest site. I think this has to do with provisioning and defensive allocation. I think that the seedlings that are in the wettest sites are generally better defended against pathogens and therefore have a decreased risk of pathogen infection. I also found this interesting relationship with time. With each census, there was a higher probability of foliar disease. And I think this is consistent with exposure period. And as seedlings are exposed for longer periods of time to disease, they are more likely to develop disease. I found only marginally non-significant evidence for density-dependent pathogen infection, and I did not find any evidence for distance-dependent pathogen infection. There was a relationship between dispersal mechanism and probability of infection. For species that were dispersed by birds, they had a decreased probability of disease, but seedlings that were dispersed by non-flying mammals had an increased probability of disease. They were actually dispersed about the same distance, so it wasn't a dispersal distance that could explain this. However, the non-flying mammal dispersed species were heavier seeded than those that were bird dispersed, so seed size might play a role here. Overall, I found that most seedlings appear asymptomatic of infection. Mean annual precipitation and dispersal mechanism influence disease prevalence in seedlings, and this might be explained by defensive allocation or seed size, but I need to do a little bit more work to have a potential answer for you. And foliar pathogens may act in a density-dependent manner, but more data is needed before I can really feel confident in those conclusions. Additionally, data on damping off disease is needed to fully understand the role of pathogens to seedling community dynamics, but that would also require lots of intensive observations in the field sites. I would like to thank all of the people who have helped me with this work, in addition to all of my funding sources, and I look forward to your questions.
Well, thank you all. That was just fantastic. Some just remarkable stuff being done. I was trying to take notes the whole time. I have questions of my own. I thought it was really, really great, and I'd like to thank you for that, and thank you for your patience here. I know there's some exciting sessions coming up afterwards, and of course, award session, and it wouldn't be ATBC without the party and banquet, which is, let's face it, one of the things we're known for. So I do want to get to some questions though. And again, if people don't have any questions now and would like to come back and post them later, they're welcome to do so over on the page for this session. And if they have any questions now, do that. So I've got some questions here that either were some I ended up getting, someone sent them to me by phone, and then I also have some of my own. So I'll try to ask those, and if anybody has any questions as they come up, we've got a couple minutes left, so feel free to ask. The first question here is for Flavia, actually. Is Flavia's still here? Yes. So Flavia, the first thing is I was just going to say that 50% of those 224 species being singletons is just amazing. That's outrageous. But that's actually not my question. That's just my kind of one of the mind-blowing facts I got out of today's session. I was really interested in the phytochemistry and its seasonal variation. And if you could tell us a little bit more about that variation. I was trying to take notes, so I may have missed some of the details. Was that variation in that non-overlap, was that in the abundance of compounds, the different compounds, carbon-based versus nitrogen-based? Was it about constitutive defenses being more common in one season and induced defenses being more common in another? Can you tell me a little bit about the way in which they were different?
Yeah, can you hear me?
Yeah, perfectly. Thank you.
Okay. Thank you for your question. We are not working on compounds because this is NMR data. So we're still working on the identification of compounds. But when I tell you that they are different, it's just because of the spectra. They're different. So the spectra, the different peaks of the spectra means some kind of a chemical function. So we know that maybe the compound is similar, but it has some differences. So what we see is that the structure is the same, but it gets some partial change across the seasons. So maybe there's just small differences in the compound, but we don't know the compounds yet. So it's just the difference between this chemical functions that change across different seasons.
No, thank you. That's great. That's exactly what I was hoping to hear. That's really fascinating. Let me see. The next question that came across here was, I'm gonna just take advantage of the fact that I have the microphone, and I mean, I suppose Javi could cut off my Zoom, and mute me if she wanted to, because she's in charge. But I'm gonna take advantage of asking my questions too. This question is for Danelka. I confess, it had never crossed my mind to think about these exotic gastropods like that. And I mean, I've read lots of papers on, edge effects and disturbance and invasive herbivores, and I think this may well be the first time that I'd seen it and seen the abundance and seen the number of invasive species on these edges and in these disturbed habitats. Do you know if this has been documented other places? Is it that people have looked for exotic gastropods in these kind of disturbed areas or forest edges or something and haven't found them? Is it that nobody's bothered to look before? I thought this was so cool.
Hello, I'm Dinelka. Can you hear me?
Yes, thank you.
Actually, when I'm doing my MPhil, I was able to found that some of these gastropods in Egypt and some other tropical regions, there are some few forest invasions there. So that's why I use these terms to explain those things. And during my study, I was able to found that some of these exotic gastropods, especially this invasive one, the Lysacatina fulica, the giant African snail, it is a huge threat to the Sri Lanka. Not only in the tropical rainforest, but they also found in the dry zone forest in Sri Lanka as well. Basically, I found those things were invade in the buffer zones, but some of them were able to successfully invade the core forest as well.
That's just amazing. I had never thought of it. The next time I go into the field, I'm gonna have to get over my feelings about gastropods, and I'm gonna grab everyone I find and see if someone can help me identify it. It reminded me of maybe another one of those cases where if you were to ask like the farmers if they had problems with gastropods eating, all their plants, they would say, of course we do. We spend all this, we lose lots of our crop to them. And then you ask the ecologists who work in the reserve next door, do you have anything, do you know anything about exotic gastropods? They go, yeah, it's not really an issue here. So this was really insightful. I thought it was really interesting. And so I'm gonna go and look up more. This is fantastic.
Actually, Sri Lanka also have the same problem because most of these agricultural lands are closely adjacent to the natural forest. That's why I think these invasive species can invade it.
Yeah, and in fact, I found that to be a really remarkable theme throughout the session here, which was that this really important role of the interface of ecology and agroecology and the potential for invasion and things coming through. We've got a couple of talks here. Hey, Camila Pisano has a question here for Antonio. You mentioned at the end of your talk that it is very hard to find native species seedlings for restoration. I would like to know if you managed to develop or share protocols for planting and including species you found to be key for hummingbird restoration projects in Ecuador.
Thank you, Camila. Thank you, everyone, for those wonderful talks. Yes, it has been some work. Actually, my lab has an attached plant nursery, and we do a lot of research on those species in particular. So I would say we have around 20 species that already we have protocols known, and we have some results on field experiments. So there's a lot of work that's being in progress right now. Unfortunately, none of it is published yet, but we hope to do some work on that. So yeah, it's one of our priorities. Thank you.
Excellent, thank you. This next question that's come through is for Valerie. Are the dry sites or species at the dry sites historically as dry as they are now? Michelle, who asked the question, is wondering if one of the reasons you're seeing higher disease prevalence in the dry site seedlings is because they are not adapted for climate change conditions. I had kind of a related question, which is it potentially that the dryness makes them more susceptible because of stress? But those are different questions about the same aspect. So let's start with Michelle's first, which is they're not adapted for those particular conditions, perhaps.
So the dry sites the driest site that I surveyed still gets about 1,800 millimeters of precipitation annually. So it is still quite wet. It's just relative to that gradient, which maxes out at 3,500 approximately millimeters per year. That's the dry site relative to the wet site, or the wettest sites. So it's still quite wet, and I do not believe that it has become much, much drier with climate change, at least so far. Although we do expect to see like an increase in like seasonality or seasonal differences where like the dry season becomes drier and the wet season becomes wetter with climate change. But so far that has not been, we've not seen that yet, if that answers the first question. Okay, and then can you repeat what your question is?
I was just wondering about the potential for drier sites being more stressful and therefore making them more susceptible. But it sounds like, drier here is kind of a relative term anyway, so maybe we're not at that point. But
yeah, but that was actually something I was very interested in, especially into the dry season, because then, those seedlings are starting to become stressed. And there is a possibility that being stressed because those seedlings are now in drier conditions, like, yes, they are more susceptible for the development of symptoms. And so there is a possibility that that is partly why I saw an increase in the amount of disease in the dry season as time was passing. But it's really hard to disentangle that from the fact that like over time with each census, seedlings were more likely to become diseased.
Excellent, thank you. Let me see, we have a couple of minutes left here, and I know we have at least one other question. Before we get to that one, I'd like to ask one here of Serena. Is Serena still? Yeah, yes, good, you're there. So again, this is another one of these talks that I've seen over the last bunch of days that has kind of just, this was so cool, this idea that you have this natural stratification of fruit resources, just awesome. And so my first question was, is this something that this was a really great case study? And so my first question was, are there other things that this could be done with? So is that kind of either, like liana style where it grows down all the way up into the canopy with fruit at all levels rare? Is it common? Are there other ones? I thought about maybe coniferous trees, where the fruits go straight from the trunk. So maybe that's another opportunity to test those. But that was just like ideas. The real question I have is, this, and it's kind of a weird question, so bear with me. This is a really cool thing in that you found that you had these different relationships between the obligate and the more generalist frugivores, right? And some of these obligates might be better dispersers, more effective dispersers, or maybe long distance dispersers than some of the generalists. But maybe the generalists, maybe they're more abundant or they consume more. And so what it really had me thinking is that you might have different conflicting selection pressures depending on where on seeds. You might think to have selection for dispersal by the things that are up in the canopy, and yet, those kinds of fruits aren't going to do you any good in the understory, and maybe the reverse. So this is not just kind of, I think, interesting from the ecological aspects, but it really says something maybe, or maybe I'm wrong, about the selection pressures and evolution for fruit characteristics in plants like this. Is that possible? It seems like it seems like there should be selection for whatever is going to be the most effective disperser, right? It's just there's this weird trade-off there I can't figure out. I think it's so cool. It seems like a real puzzle.
Yeah.
You can tell me I'm wrong. It's okay.
No, I mean,
you wouldn't be the first. Javi will tell you. You wouldn't be the first.
I mean, and normally it is like that. I mean, you were also right. There are not many plant species who offer their resources across all strata. So, I mean, there are some like liana or cauliflower species, but not many. And normally there is a lot of selection happening also between strata, and the fruits just look different. So, I mean, there are also many papers showing, for instance, that fruits in the canopy are bigger compared to those who have their fruits in the understory. But yeah, I can only say that our species is endemic in this area and it's super abundant there. So compared to all the other species, it's really, really abundant. So it somehow at least seems to be kind of effective. So maybe that's just the selection that's happening here. The fruits are small and
in all the strata, the fruits are small.
They're small in all the strata, but but by offering them across all strata, there's still enough birds coming, or bats to disperse them.
Great,
so
maybe it's just
another
that
kind of
intermediate
trait.
right size. Yeah, it could be. Yeah, I suppose that's right. That's cool. Well, we have time for one last question, and Camila Pisano gets the last word. She usually does. And so Camila's question here is, I have a question for Valerie. Did you uproot any seedlings to look at below ground pathogen effects? Do you suspect any of the seedlings you found in the field were the survivors of below ground pathogens, or do you think below ground enemies didn't really have a major evident effect in your study?
Camila, thank you for that question. No, I did not uproot any of my seedlings to check for any sort of like attack on the roots. I do, however, think that those interactions are incredibly important. I actually think that that was something that, given my survey method, it was very difficult for me to observe. And that's the kind of thing that I would love to go back and really like take a very long and intensive look at the seedlings like over time to watch like how maybe a specific patch is being attacked over time, to understand really the degree to which those below ground pests are attacking seedlings. But I think they're very important. I just was not able to observe them with my method.
Thank you, Valerie. And that brings us to the end of our session. I want to thank our audience for their questions and their participation. I want to obviously thank our speakers. Really great stuff, really thought-provoking. Don't forget, we have in about 10 minutes, I think it is, the plenary session on diversity, equity, inclusion, lessons learned from the Workplace Environment Survey. We have our closing. We have some amazing social activities, music, everything from traditional Mexican music to electronica. So we've got prizes and awards. Who doesn't love those? We'll see you all there. Okay. Thanks, everybody.